Hi, I'm Chris Buckley. I'm Kennedy Professor of Translational Rheumatology, and I work at the universities of Birmingham and Oxford. And I've just come from a session where we were looking at the use of single-cell sequencing, single-cell analysis, to transform the way in which we think about how rheumatology is classified and managed, rheumatological conditions. Here's the problem. At the moment, medicine is classified anatomically. So you have rheumatologists, gastroenterologists, dermatologists, and that stood the test of time for the last 20 years as we've tried to get therapies forward. In the last 20 years as well, there's been a huge advance in our understanding of the genetic basis of disease based on DNA. But there's a problem because genes don't make organs. Genes make cells that make organs, and understanding the cellular basis of disease is a problem. So if I said to you, how many individual cell types make up a joint? How many individual cell types make up a brain or a gut? We don't know. So there's a huge chunk missing from the atlas that forms the joint. We know there are certain things like tendons, cartilage, synovial tissue, but what's the complexity within that? And so the advent of this ability to basically take single cells and analyze not only how many there are, but what they do, will really revolutionize the way we have the taxonomy of these diseases, because we can say this is a disease of a particular cell type, which we will want to now understand how the genes and the proteins involved in it work. Let me give you an analogy. When you wake up in the morning, you have a bowl of fruit, right? In that bowl of fruit are lots of different types of things, apples, oranges, pears, bananas, and you make a smoothie. In that smoothie, you can taste many things, but actually you don't know what the individual components were. So you're trying to impute from the smoothie how many different bits of fruit were in it. You might guess, but you won't know for sure. Single-cell sequencing takes that smoothie and allows us to deconstruct it back to the individual types of cells. So you know you've got 5 strawberries, 4 raspberries, 10 bananas, right? And the power of that means we're in a completely different way of doing medicine.